Hey team. All right, so I'm going to walk through this worksheet and kind of highlight certain things about the model. I'm not going to go through and answer the questions one by one because that's your responsibility, but hopefully you find this helpful in general. So in terms of our starting cell, remember there's an emphasis that this is the starting cell in sex organs and that the DNA has been duplicated. The reason you know the DNA has been duplicated is because you have X's. So if you remember here, this is one sister, and then this right here is the other sister. So we know that at this point the DNA has already been duplicated. A couple other things to point out in this image, in case you were a little bit confused. I think what they're doing is they're trying to show, like, say, gray is mom and the darker is dad or something like that. And this is chromosome, say, number 1, and this is chromosome number 1. So this is the homologous pair. So these are number 1 homologs, okay? And then down here, let's say this is chromosome number 7. So those are the two chromosome number 7s. So this would be the homologs of chromosome 7. Now, as you are thinking about this, right, it is really, really important to remember, again, purpose of meiosis is that we want a cell at the end that has exactly one version of either homolog. So you're not going to have both homologs, unlike in mitosis. Okay, so as we're going through here, it's super, super important just to keep in mind that we're literally looking at different chromosomes, and that you would have, in terms of at this metaphase 1 right here, it's very important to remember that you would have 23 of these pairings. Right, because the whole idea is to have 1 to 23 and 1N of them in the final cell. So you match it up. So right here, this is 2N, right? Because here's the homolog from dad, here's the homolog from mom. And they code for the same things, but they are not technically identical. Whereas the sisters are supposed to be identical. So that sister is supposed to be essentially identical with that sister. All right. These two are homologs, but they are not identical. And so what's going to happen in metaphase is you're going to have this kind of, think about it as a line here, and the homologs are going to line up and they're going to match up. That way one homolog goes each direction, because you want to make sure, again, you have 1 to 23, one of each of those in the ending cells of meiosis 1. So as you can see here, the homologs are separated, but there are still two versions, right? So each of these cells, there are still two copies of this information because of the two sisters. So we're not done yet. So pulling the homologs apart is really important, and it allows these guys to be 1N, though they're 1N duplicated, right? It's 1N because you only have one version of the chromosome, because again, the sisters are identical, so they're technically the same version of the chromosome. All right, so hopefully that kind of helps in terms of some of the answers there. In terms of this, alleles. Alleles are going to be essentially different versions of a trait or a gene. So if we say the gene is hair color, your alleles would be brown and blonde and red, essentially, would be your alleles. To varying degrees, it gets a bit more complicated, but that's the idea. So if you get one chromosome from one parent that's brown and the other that's blonde, those two chromosomes, right? Let's say this is on chromosome 13. I actually don't know what chromosome it's on. Those two chromosomes are homologs, but they're not identical, and we know they're not identical because if one's coding for brown hair and one's coding for blonde hair, then they can't possibly be identical. So just a heads up in that sense, like that when we talk about alleles, we're talking about it's going to be located in the same place of a chromosome, but the DNA is not going to be exactly identical. Let's see. I think that's good. That's good. I'm just going through kind of some of the terms. Diploid is essentially, think about it as versions of the chromosome. So most of our cells are 2N because dad and mom. Haploid are going to be the gametes, and that's because you want exactly 1/2 of your chromosomes. So if you think about it, this is like kind of one of my favorite mental games, is that in reality, you are essentially giving the offspring egg or sperm, okay, you're essentially giving them some combination of your parents. And what I mean by that is your chromosomes, you have one set of mom and you have one set of dad. And so of these two sets, you're going to give completely random, like you might give the mom number 1 and the dad number 2, etc., etc. You're going to give completely random of them to the gametes. So the gametes, right, this egg or sperm cell, are going to be essentially some combination 1 to 23 of either your mom or your dad, right? So this is where sometimes you can see things like, quote, skip a generation, where one of or you might look more like your grandparents or something, more like one of your grandparents or something like that. And that can be why, because it is possible that you get more genetic information from, say, your grandfather than your grandmother, or vice versa, which is really, really nifty. Because when we think about reproduction, it is generational, right? So you're constantly you're not just reliant on this current generation, you're reliant on the generation before and before and before, because where do your parents get their DNA and their chromosomes, etc., etc., and you can keep building. In terms of meiosis too, this one is very simple and it looks much more like mitosis in that sense. The difference is that in mitosis, you would have the second homolog, right? So you would have, say, this is dad's chromosome, and then you would have mom's. But we don't have that in meiosis, right? Because again, we're starting with haploid here, and then we're just pulling the sisters apart. So now at the end, when I have my ultimate cells here, I have 1N non-duplicated, right? So I have one version of 1/2 of my starting DNA, if that makes sense. I hope that makes sense. Um, and it could be random, and the thing about it is this also could have broken apart into a different way, right? You could have gotten this chromosome with this chromosome and vice versa, this chromosome with this chromosome, and that's all part of the randomness, like how they all decide to align. And that alignment, if you think about it, takes place right here. So meiosis, or excuse me, metaphase 1 is really when the kind of determination of all the different variation occurs. Okay, because that's when the homologs line up. You could have dark and dark on this side and light on this side, or mixed. And then remember, that's happening for 23 of them. So there are technically, for my math people, 2 to the 23rd variations of just how those homologs line up and divide, which is kind of bonkers to think about. All right, coming down here. So this is essentially talking about what it ultimately looks like in terms of fertilization. So now we're really getting to that reproduction. So we have a single sperm, which is going to carry, oh, that's not what I was trying to draw, is going to carry 1 to the 23rd or 1N chromosomes. And then you have an egg, which is going to carry 1N chromosomes. I don't so much need you to worry about polar bodies, but we can talk about it. It's very, very interesting and cool. But the idea is you have organelles in here, in the egg, that you don't have in the sperm. So the egg cell needs to have all the starting stuff, the mitochondria, endoplasmic reticulum, etc., etc. So in terms of what it does is it actually takes the good stuff from one of the oocytes, and it essentially takes that, leaves the DNA, and it degrades elsewhere so that the egg can be super large. So the egg has all the resources needed. And so the sperm comes in, all the sperm brings to the situation is the DNA, that 1N, and now we have a 2N zygote, which will inevitably be, right, the offspring. And remember, this is happening 1 to 23. So 1 to 23 from dad, 1 to 23 from mom. Okay? terminology, ploidy. Ploidy is essentially how many sets of chromosomes. Sets of chromosomes. All right, so if you have a set from mom plus a set from dad, you have 2 sets of chromosomes. If you have broken this apart so that you have half of them, you have 1 N of chromosomes in that sense. If we didn't do this process of meiosis and you had a 2N match with a 2N and they give all of their chromosomes to the offspring, it's a 4N. And then what if that 4N matched with another 4N? Now all of a sudden, right, you have 8 N, and so on and so on and so on. So this meiosis process is to prevent all this chaos with chromosomes so that we always, as individuals, have 2N, 1 set from mom, 1 set from dad. All right, and finally, this image is where things get complicated, but in all honesty are super, super interesting. So I want to really go back to this idea that meiosis, the objective is twofold. One is to create gametes with 1N, right? Half of the DNA, and 2 is genetic variation. Those 2 things are absolutely imperative so that we go from 2N to 1N so that it combined with another 1N and bring us back to 2N, and genetic variation. Now you got a lot of genetic variation up in meiosis 1. So meiosis 1, you have the term what we call independent assortment. And essentially what happened there, essentially what happened there is that the 1 to 23s lined up in some way. So say this was mom and this was dad, and then number 2, say it was dad and then it was mom, and then number 3, dad and mom, and number 4, mom and dad, all the way to 23. And this is completely random. So the way that this lines up is completely different from the way that this lines up, which is why we say independent. So the different homologs line up independently of one another, okay? And then what's going to happen is that those homologs are going to go that way, those homologs are going to go that way, and there you have, that's where we get the 2 to the 23rd variation because of all the different ways that those chromosomes can line up. So already we're talking it would be nearly impossible. That's like 0.00000 chance that they could line up identically, and you could have essentially this like individuals who have exactly the same DNA. But as all good things in biology, the next most important thing is genetic variation. And so there's a way to ensure that it is impossible, actually impossible, to have an identical, 2 identical sperm or egg cells, 2 identical gametes. And that's through crossover in metaphase 1. So in metaphase 1, when we are in our tetrad, the inside sister, so again, I'm going to draw this because I think it's helpful. So this is one sister here, right? And then this is another sister here. And so what will happen is that the inner sisters, so this sister right here and this sister right here, will cross over and exchange genetic material and just do a clean swap. So we're cutting at exactly the same place and just essentially swapping DNA. But think about what that did. So now let's say this was dad. You have a dad sister, boom, boom, that is 100% dad's chromosome. You have a mom sister that is 100% mom. But then in the middle, look at this. You have these 2 chromosomes that we call recombinant because we have recombined the DNA. And so what happens is these are mixed. So you can be giving your offspring some combination of mom and dad's DNA in that chromosome, which is so cool. And essentially what this does is this creates limitless genetic variation because you have all this happening 1 to 23. So which cell is going to get mom or dad's initial like chromosome or sister chromatid, and then on top of that you have all this crossover, which means you've created all these new chromatids that have never existed before. And so there's also the possibility to get those. So from a statistical standpoint, it is basically at this point impossible to have two genetically identical sperm or egg cells, and therefore it is impossible to have genetically identical offspring. So I hope that you found this helpful. I'm just going to scroll through here. I hope that you found this helpful. I think I've addressed all of the major terms and whatnot. So finish this worksheet. That's the task. Finish the worksheet and then bring your questions to class on Friday. I hope you found this helpful. Talk to you guys later. Bye-bye.